Right now, inside the mouths, the digestive tracts, and the mucosal linings of probably up to 70% of everyone listening, there is a microscopic stowaway.
Yeah, and it's an organism that completely shatters the traditional way we are taught to think about disease.
Exactly. I mean, we usually picture illness as a breach, right? A hostile microbe from the outside world invades the body, and the mission is simply to expel it. But what happens when the invader is, well, already living quietly in your spare bedroom?
It forces a total reevaluation of what constitutes a threat. We're looking at a master of adaptation today. It's an organism whose behavior depends entirely on the delicate, invisible ecosystems keeping us alive every single day.
Because the hostility doesn't come from the microbe itself. It comes from sudden shifts in the environment we provide for it.
Right, exactly.
So, welcome to today's deep dive. We are putting Candida under the microscope. You likely know it as the fungus responsible for yeast infections or oral thrush.
Which is incredibly common, yeah.
Very common. And we have gathered a substantial stack of sources today. We're pulling from microbiological textbook definitions, clinical literature reviews, and even historical taxonomic accounts.
It's a really comprehensive look.
It is. Our mission for this deep dive is to fundamentally reframe your understanding of this organism. We are going to explore how a harmless resident transforms into an invasive threat. We'll unpack the pervasive internet myths surrounding it and look at a terrifying new strain currently challenging global health networks. Okay, let's unpack this because the underlying mechanics of this fungus are just incredibly complex.
They really are. And to truly understand Candida, I think we need to strip away the clinical assumptions and look at its base taxonomy. Candida is a genus of single-celled fungi, essentially yeasts.
Right.
There are roughly 200 recognized species within this genus. Yet out of those 200, fewer than a dozen regularly cause disease in humans. The dominant player, the one responsible for the vast majority of human infections, is Candida albicans.
Yeah, and the standard microbiological textbook definition of Candida albicans is a bit of a mouthful. It describes it as an opportunistic, dimorphic fungal pathogen that is also a normal commensal of human mucosal surfaces.
That is a very dense string of terms.
Right. But looking at the clinical reviews, the word commensal seems to be the one that completely upends our traditional model of infection.
Oh, absolutely. It is the cornerstone of the organism's success. Being a commensal means Candida albicans lives in harmony with the host. It resides quietly in the gut, the mouth, and the vaginal tract of the majority of healthy humans without causing a single symptom.
It's just hanging out.
Exactly. It is an expected native member of the human microbiome, which is the fungal counterpart to our bacterial microbiome.
Okay, I am trying to wrap my head around the terminology here, though. If this fungus is completely harmless in up to 70% of us, simply acting like a quiet roommate who just keeps to themselves when the landlord is around, should we even be classifying it as a pathogen at all?
This raises an important question, and it really strikes the heart of a major debate in modern microbiology. For over a century, infectious disease was largely governed by Koch's postulates.
Right, from the 19th century.
Exactly. That late 19th century framework stated that a specific microbe causes a specific disease in every host. It's a one-to-one relationship. You encounter the cholera bacterium, you get cholera. The microbe is the villain.
But Candida doesn't fit that model at all. You can be colonized by it your entire life and never get sick.
Never. And that's why in the 2010s, researchers like Bernhard Hube and Julian Naglich really helped shift the scientific consensus regarding organisms like this. Virulence in Candida is not an intrinsic property of the fungus in isolation. It is an emergent property of the host-microbe relationship.
Oh, I see.
Yeah, the label opportunistic pathogen is highly contested, precisely because it only inflicts harm when the host's internal environment provides the opportunity.
So, mechanically speaking, what actually constitutes an opportunity for a single-celled fungus? I mean, if it's normally just taking up space, what flips the switch to make it attack?
Well, it is an ecological numbers game. Your mucosal surfaces are not empty terrain. They are dense, highly competitive ecosystems. In the gut and mouth, hundreds of bacterial species are constantly competing with Candida for nutrients and physical attachment space.
So the bacteria are keeping it in check.
Exactly. And on top of that, you have host defensive saliva, stomach acid, and a highly specialized mucosal immune system. Specifically, we rely heavily on Th17 cells.
Right. I've seen Th17 cells mentioned all over the literature alongside IL-17 signaling. But how do they actually interact with the fungus?
Think of Th17 cells as a specialized border patrol for your mucosal surfaces. They are a type of T cell that constantly surveys for fungal overgrowth. When they detect it, they release chemical signals called interleukin-17, or IL-17.
Okay.
This signal recruits other white blood cells, primarily neutrophils, to the area to swallow up the excess yeast and just keep the population at a safe baseline level.
Wow, okay, so it's like a mature forest floor. Every inch of soil is occupied by native plants. The bacteria and the immune system is actively pruning anything that overgrows.
That's a great analogy.
But if a child takes a broad-spectrum antibiotic for, say, an ear infection, you are essentially causing a forest fire. You wipe out all the native bacterial plants, leaving empty soil and abundant nutrients.
And Candida, which is naturally resistant to antibacterial drugs, just survives the fire. Without bacterial competition, it blooms.
That makes total sense.
Conversely, consider a patient undergoing chemotherapy. Their white blood cell count plummets. The Th17 border patrol is entirely removed. The bacteria might still be there, but the immune suppression allows the fungus to proliferate unchecked. Mechanistically, these are identical failures of the body's control system, just originating from opposite directions.
And here's where it gets really wild, because when those controls fail, Candida doesn't just divide and increase its numbers, it physically alters its entire morphological structure. This is the dimorphic aspect of that textbook definition we mentioned earlier.
Yes, though some mycologists actually prefer the term polymorphic, because Candida albicans can cycle through several distinct forms, including a highly resilient resting phase called a chlamydospore.
Oh, interesting.
But the primary, most critical transition is from yeast to hyphae. In friendly, crowded conditions, the organism exists as a round, budding yeast cell. In this form, it is really easily engulfed and destroyed by roaming immune cells like macrophages.
But the sources indicate the fungus is constantly sensing its environment, right? Like when it detects human body heat at exactly 37 degrees Celsius, or a neutral pH, or even the presence of serum proteins from our blood, it stretches out. It literally germinates into these long filamentous threads called hyphae.
What's fascinating here is that the yeast to hyphae transition is one of the most rigorously studied virulence mechanisms in medical mycology. Those filamentous hyphae act as microscopic spears. Yeah, because they are elongated, immune cells can no longer easily swallow them. They're just too big. The fungus uses these filaments to apply physical mechanical pressure to pierce human epithelial tissue and invade deeper into the mucosa.
Wait, I need to push back on that slightly. Is it purely mechanical? Because the literature from Julian Naglick's group in 2016 mentions a chemical component as well. It's not just a physical spear.
You're completely right. The physical piercing is only half the strategy. Naglick's group discovered that as the hyphae invade, they secrete a highly specific peptide toxin called candidiasis.
A custom toxin.
Exactly. This was a monumental discovery because it is the first classic peptide toxin ever identified in a human fungal pathogen. Candidiasis directly embeds itself into the membranes of our epithelial cells, forming pores that basically rupture the host cell, releasing nutrients for the fungus to absorb.
So it shapeshifts to become too big to eat, physically stabs the tissue, and secretes a custom toxin to burst the cells open.
It is brutal microscopic warfare.
The structure and shape seem completely integral to the attack. I was looking at research from Alexander Johnson's lab at UCSF and Neil Gow's in Aberdeen. They genetically engineered mutant strains of Candida, and some mutants were permanently locked in the round yeast form, while others were permanently locked as long hyphae.
And neither mutant effectively caused disease, right?
Exactly, neither of them. The virulence isn't inherent to either shape itself. It's the ability to transition between them in real time that makes it deadly.
It is the ultimate adaptability. By constantly shifting its morphology, it evades the immune system, penetrates tissue, and extracts nutrients. But to see how this microscopic warfare translates to clinical reality, we have to look at the visible manifestations, the tip of the clinical iceberg, so to speak.
For most people, this organism is synonymous with oropharyngeal candidiasis, which is commonly known as thrush, or vaginal yeast infections. Thrush presents as these creamy white plaques on the tongue, inner cheeks, and throat.
And the sources point out that scraping those plaques reveals raw, bleeding tissue underneath.
Which makes perfect sense now that we know about the hyphae physically puncturing the cells and the candidiasis rupturing them.
Right. And thrush is particularly prevalent in populations where that ecological balance we discussed is fragile. Newborns frequently develop it because their native bacterial flora and mucosal immune systems are just not fully established yet.
So it's not about age, it's about the microbial status.
Exactly. We also see it extensively in patients with asthma or COPD.
Because of the inhaled corticosteroids they use, right?
Yeah.
The steroids locally suppress the immune response in the throat. It's a direct, localized dismantling of that Th17 border patrol, which is exactly why doctors always say you have to rinse your mouth after using a steroid inhaler.
Yes. And we also see a massive prevalence in individuals who wear dentures. Studies indicate roughly two-thirds of long-term denture users develop a localized candidiasis.
Wait, really? How does an artificial piece of acrylic trigger a fungal infection? I mean, there is no biological tissue there for the immune system to fail at protecting.
Well, the denture provides a unique mechanical advantage. It creates a warm, low-oxygen pocket between the acrylic and the gums. But more importantly, it is a non-vascular surface. Immune cells travel through blood vessels. They cannot patrol the surface of a plastic denture.
Oh,
This
yeah.
allows Candida to attach to the acrylic and construct a biofilm.
Okay, I want to clarify how a biofilm actually functions here. It's not just a dense cluster of cells, is it?
No, it's a highly structured microbial community encased in a self-produced matrix. Essentially, the fungi secrete a dense, sticky shield made of carbohydrates and proteins. This matrix physically blocks immune cells from reaching the fungi and acts as a barrier that prevents antifungal drugs from penetrating.
So a denture is basically a luxury condominium for a fungus. It's an artificial safe harbor where it can build a fortified bunker.
That is a very vivid way to put it, but yes.
It perfectly illustrates how environmental changes dictate the behavior of this organism. And the same principles apply to vulvovaginal candidiasis, which affects an estimated 75% of women at least once in their lifetime.
And a significant subset, roughly 5 to 8%, experience recurrent infections. This brings us to a major clinical misconception that really requires correcting. The belief that vaginal yeast infections are a sexually transmitted infection.
Right. Based on everything we've discussed about the native microbiome, classifying it as an STI fundamentally misunderstands where the fungus even comes from.
While sexual activity can alter the local environment, the infection is overwhelmingly driven by the disruption of the individual's own existing flora. Fluctuations in hormones like estrogen, elevated blood sugar, and uncontrolled diabetes, or just courses of antibiotics disrupt the local vaginal ecosystem.
The candida is already present. The environmental disruption simply removes the checks and balances.
Exactly. Now, those mucosal infections are common and treatable, but the clinical literature reveals a much more severe hidden danger when Candida breaches the mucosal barrier entirely and enters the bloodstream. Invasive candidiasis or candidemia.
Candidemia is a severe medical emergency, primarily occurring in hospital settings, right?
Yes. The fungus typically gains access to the bloodstream via central venous catheters. These are the specialized IV lines placed in the chest or neck of critically ill patients.
Another artificial surface.
Exactly.
Yeah.
Once in the blood, Candida can seed itself in major organs: the kidneys, the heart valves, the eyes.
And the mortality statistics for candidemia in intensive care units are just shocking. They're routinely cited between 25 and 50%. We've developed incredible medical interventions like chemotherapy, central lines, broad-spectrum antibiotics. But by keeping critically ill, immunocompromised patients alive, we have inadvertently designed the optimal ecological niches for this fungus.
It is a stark paradox in modern medicine. Our most advanced life-saving techniques simultaneously dismantle the host's natural barriers. We are providing a native commensal with an unimpeded path to vital organs.
The sheer prevalence of Candida, coupled with its ability to cause so many diverse localized issues, has made it a massive target for misinformation and alternative medicine narratives, too.
Oh, absolutely.
Since the 1980s, there has been this pervasive concept circulating online called Candida overgrowth syndrome.
Right. The central claim of this fake syndrome is that a hidden systemic overgrowth of Candida in the gut is responsible for a wide array of nonspecific symptoms. Proponents blame it for chronic fatigue, brain fog, joint pain, depression, and even autism spectrum behaviors.
I'm really confused by how this narrative gains so much traction. I mean, if candidemia in the blood is a critical ICU emergency with a 50% mortality rate, how can someone claim to be walking around for years with a systemic Candida overgrowth causing, like, brain fog?
The confusion stems from conflating localized gut colonization with systemic disease. Candida does live in the gut. It can overgrow in the gut after antibiotics. However, Mainstream mycology, which is supported by extensive reviews, including a definitive 1990 report in the Journal of the American Medical Association, found absolutely zero rigorous evidence that this localized gut overgrowth causes the sweeping systemic neurological or rheumatological symptoms claimed by the syndrome.
Yet the proposed solution online is always a strict Candida cleanse diet, demanding the elimination of all sugars, yeast, and carbohydrates. It's treating the fungus as an absolute evil that just must be purged from the body. It's so insidious because they took a real biological fact. Yes, yeast feeds on sugar and can overgrow, and wrapped it in a fake internet diet culture narrative to sell cleanses.
And it provides a perfect teaching contrast for students on how to separate well-documented disease from plausible-sounding medical myths. The medical objective with a commensal organism is always restoration of balance, never total eradication. The idea that Candida is always bad and must be eliminated is just false. Eradicating Candida from a human host is neither practical nor biologically sound.
And this misinformation isn't isolated to internet diets. It impacts how people approach actual diagnosed yeast infections. There is a really dangerous common assumption out there that any Candida infection can be resolved with a standard over-the-counter antifungal medication like fluconazole.
Operating under that assumption in a clinical environment is highly dangerous. As we established, there are multiple species of Candida, and they possess vastly different drug susceptibility profiles. Consider Candida krusei. This species is intrinsically resistant to fluconazole.
How does that intrinsic resistance actually work at a cellular level? Like, why does the drug just bounce off?
Well, fluconazole works by inhibiting an enzyme the fungus needs to build its cell membrane. Candida krusei naturally possesses a mutated version of that target enzyme, meaning the drug simply cannot bind to it.
Oh, I see.
And additionally, it utilizes specialized proteins called efflux pumps in its cell membrane. So even if the fluconazole manages to get inside the cell, these pumps physically spit the drug back out before it can act.
So prescribing fluconazole for a C. krusei infection is essentially administering a placebo.
Exactly.
This highlights why identifying the exact species is not just some academic exercise. It literally dictates the survival of the patient in severe cases.
Hospital laboratories have to utilize sophisticated diagnostics to differentiate these species. They might use chromagar, which is a specialized culture medium containing enzymes that react with different Candida species, causing them to grow in distinct, identifiable colors on the plate.
That's pretty cool.
Yeah, or alternatively, they use Melditoff mass spectrometry. This involves using a laser to vaporize the fungal proteins, creating a highly specific mass spectrum fingerprint that identifies the exact species in minutes.
Which means we rely entirely on our taxonomy, our system of classifying and naming these organisms. And the history of naming this fungus is surprisingly messy. It wasn't just discovered and named overnight.
Not at all.
In 1839, a German physician, Bernhard von Langenbeck, first observed the fungal filaments under a microscope in a typhoid patient.
But for decades, it lacked a unified classification. In 1853, Charles Philippe Robin named it Ordium albicans. By 1890, Wilhelm Zopp reclassified it as Manillia albicans, a name that actually persisted in the medical community well into the mid-20th century.
Right. And the modern name we currently use, Candida, didn't even appear until 1923. A Dutch botanist, Christine Marie Burkhout, proposed it in her PhD thesis. She observed the unique way the organism budded and determined it required its own distinct genus. She selected Candida, derived from the Latin candidus, meaning white or glowing, referencing the distinct white plaques seen in clinical infections.
It is a great reminder that scientific naming is a social process, often decided by committee. Her accurate classification wasn't formally adopted until a vote at the 1954 International Botanical Congress in Paris. Naming these organisms is a rigorous process, essential for ensuring clinicians worldwide are treating the exact same pathogen.
But fast forward to recently, and a new name has emerged that is challenging our entire understanding of fungal resilience, Candida auris.
The emergence of C. auris is one of the most pressing mysteries in contemporary infectious disease. It was first isolated in 2009 in Tokyo from the ear discharge of a patient, hence auris, which is Latin for ear.
Right.
Retrospective analysis found it in South Korean blood samples from 1996. But in the 2010s, it seemingly emerged simultaneously on multiple continents, South Asia, South America, and Africa.
And it does not behave like Candida albicans, does it?
Not at all. It is frequently multi-drug resistant. We typically rely on 3 main classes of antifungal drugs, and some strains of C. auris are resistant to all of them.
That is terrifying.
Furthermore, typical Candida species do not survive long outside a host. C. aureus, however, sheds onto hospital surfaces, bed rails, medical equipment, and survives for weeks. It is resistant to many standard disinfectants, behaving much more like a hardy environmental bacterium than a human commensal yeast.
Which is why the CDC designated it an urgent threat in 2019. So what does this all mean? How does a previously unknown fungus suddenly appear globally with such an aggressive, resilient profile?
If we connect this to the bigger picture, one of the most compelling theories comes from mycologist Arturo Quesadoval. In 2019, he proposed that Candida auris may be the first pathogenic fungus to emerge as a direct result of climate change.
Wait, climate change. Let's break down the mechanics of that. Fungi are generally environmental organisms that thrive in cooler temperatures, right?
Yes, the vast majority of environmental fungi simply cannot survive at the baseline human body temperature of 37 degrees Celsius. Our internal heat is a profound evolutionary defense, known as the thermal barrier. It is a primary reason mammals thrive, while cold-blooded animals are highly susceptible to fungal diseases.
But Kestival's hypothesis suggests that as global ambient temperatures rise, environmental fungi are under selective pressure to adapt to heat just to survive in the wild.
Precisely. As these environmental species adapt to hotter climates, their heat tolerance inches closer to 37 degrees Celsius. Our mammalian thermal barrier, which has protected us for millions of years, is suddenly eroding in real time. C. auris may simply be an organism that acquired thermal tolerance in the environment, which inadvertently allowed it to comfortably colonize the human body.
And we are confronting this evolving threat with a severely limited pharmaceutical arsenal. Fungal pathogens have historically received vastly less research funding than bacteria or viruses. The most recent novel class of antifungal medications, the echinocandins, which work by inhibiting the synthesis of the fungal cell wall, were introduced in the early 2000s. We are facing a rapidly adapting organism with tools that are over 2 decades old.
It's a huge cause for broader alarm. We have relied on the assumption that fungi are mostly peaceful commensals, or that our robust immune systems and thermal barriers will just naturally protect us. But as our medical landscape shifts toward highly immunosuppressive treatments and our changing environment alters the thermal limits of the fungi themselves, those assumptions are failing.
It really fundamentally reshapes how we view the invisible world around and inside us. Let's synthesize our findings from today. Candida is not just an annoying yeast infection. It is a shape-shifting organism that utilizes complex toxins and biofilm architecture to survive. It is a native resident of our microbiome that exploits the very medical interventions we use to save lives: antibiotics, chemotherapy, and central catheters.
It's the reason over-the-counter antifungals are bestsellers globally. It requires us to view illness not as an invasion, but as an ecological collapse.
Exactly. So to everyone listening, the next time you take a broad-spectrum antibiotic or use a steroid inhaler, consider the microscopic turf war occurring right inside your mucosa. It's determining whether a quiet commensal remains peaceful or transitions into an invasive threat.
It's a delicate balance.
It really is. But I want to leave you with one final thought to mull over, building on Quesadoval's climate hypothesis. If environmental fungi are already starting to adapt to higher temperatures in the wild, and our global healthcare system is increasingly filled with vulnerable, immunosuppressed patients relying on artificial devices, are our greatest modern medical triumphs inadvertently paving the perfect, climate-controlled ecological niche for the next great fungal pandemic. Next time you think about global warming, don't just think about rising oceans. Think about the thermal barrier of your own body.